Good afternoon, all of you. Welcome, Dr. Pradeep Mishra, sir. Sir, are you ready? Can we start with the session?
Yeah, good afternoon, man. We can ready, start, man.
Okay, sir. Thank you so much, sir. Okay, hello, everyone. Respected dignitaries, eminent speakers, distinguished researchers, faculty members, and dear participants, once again, a warm welcome to all of you in this technical session of First International Conference on Research, Innovation, and Women Empowerment in Sustainable Agricultural Development. And Department of Biotechnology and AKS University welcomes you in this conference. Now it's time for our technical session of the conference, and we are honored to have with us Dr. Pradeep Mishra, sir. I would like to welcome our distinguished expert, Dr. Pradeep Mishra from the College of Agriculture, Riva, who is well known for his contributions to agriculture research and sustainable farming practices. He is having 200 plus research papers published and 2000 plus research citations also. He will give us and guide about the publication process and reputed journals and all the practices. So I will hand over the session to Dr. Pradeep Mishra. Sir, you can continue with the session. Thank you.
Yeah, good afternoon to everyone. So, first of all, welcome to this international conference on research, innovation, and women empowerment in sustainable development, which is organized by the AKS University and the Department of Biotechnology. So, first of all, I am thankful for the Higher Authority Vice Chancellor and Pro Vice Chancellor of the university and the Dr. Ashwini Madam for giving me opportunity for the deliver this lecture. So let me share my screen first. I hope it is visible to all.
Yes, sir.
Yeah.
Slide presentation view. Yes.
So actually, we are going to talk very important topic, that role. What is the role of statistical tools for the publication in research article in high impact journals? So, being as I am also the associate editor of the scientific report and the PLOS One journal. So, normally what we are seeing that when any article is coming, so first of all we are checking the plagiarism, that it is how much it is similarity is there or not. And now we are AI era, so as a author we need to be very careful when we are publishing the paper. So if we are using AI, so if we are using the AI for the language improvement also, so we have to acknowledge that thing. And we cannot use the AI more than 10% for the our whole manuscript. So we have to careful about the similarity index you are AI using. So first check that either your publication is having the how much plagiarism or similarity. So check with some authentic software like Turnitin. Okay, because the Turnitin is also using by the Springer elsewhere than big journals like that. Then the language improvement you can use for the Grammarly and Ginger, where you can already you have written. So just you can cross check that everything is good and it is in properly grammared. And sometime you can use the paraphrasing for the quillboot. But when I am saying that you can use the quillboot, it doesn't mean that you are using the AI text and just you are paraphrasing here. If you have written itself you anything, so some of the text if you are not confident, so you can just little bit correct from here. Either you can put as a creative or academic. So just for the language improvement you may take the help if you are little bit confused. Now sometime it is very confusing that where to submit the article, because most of the time people are sending me manuscript to me and they are asking that where I can or we can submit the our manuscript. So Springer is also having their journal finder, and giving the information about that what is the impact factor, what is their acceptance rate, what is their first decision time, and like that elsewhere also they are giving the information. So one thing I wanted to show before we go further, suppose I have one manuscript and we can just check what the information we are getting here. So first we will go with the Springer Journal Finder and just we put our abstract. So on the basis of the abstract, Springer suggester will give us the suggestion. So it will giving the publishing model open access. Open access means if you are having any funded, so you can publish your article in open access, but for that you have to pay by the APC, article processing charge. Hybrid mode means you can publish your paper either in open access as well as the subscription mode. Here it is giving the journal impact factor, journal first decision time. So this way Springer are giving the suggestion, and same way we can see the elsewhere also. So it is quite useful. Whatever the suggestion we are getting, it is very impactful. After that you just need to check the journal's scope and their what is their keywords. So here also it is giving either journal with the open access or journal with subscription. So here it is only open access west management bulletin, but if we go further, so like this journal is global environment change, so it is giving the acceptance date, site score, impact factor. So this way we can search our article where we can submit it. Okay, now we are coming back to our. So what is the use of the statistical tool for the publishing in high impact journal? So study design, whatever we are designing, so the number of sample size, data collection method, randomization process, we have to be very means correctly rigorous, and it should be address the our research problem. So we have to keep our study design like data analysis. Data analysis tool used that the which the research data we are using, we have to calculate their research descriptive statistics, advanced analytical technique. So we can try whatever we are using. So first we need to see the quality of the data, that whatever the data we have used, it should be good quality, and it is collected from the either primary or secondary, it should be authentic. Okay, and we have to calculate the descriptive statistic to see the nature of the data. Hypothesis is very important part, because whenever we are doing the review, so we are also giving the this kind of suggestion that what is your hypothesis, or your analysis is meeting to your hypothesis or not. So hypothesis, if it is required, so definitely kindly give into your material and method and connect with the in your result and discussion. And nowadays that the data visualization is very important when we are publishing the paper, so we have to try to prepare some different kind of charts and plots, okay, which describe our result clearly. Sometimes in instead of table we can directly use the figures also. Statistical validation is very much important, because when we are doing any analysis, so how much it is reliable or what is their validation, it is very much important. Meta analysis nowadays is very important. If you are doing single analysis for the multiple studies on a specific topic, so meta analysis making very key impact on that. Collaborate with the statistician if you are not confident about the study design, hypothesis testing, so you can collaborate the statistician also. Follow the journal guideline, whatever the each and every good journals are having the good journal guideline, so just follow them. Otherwise it will increase the chance of the acceptance, or it will go for the proper review. Always go for the updated statistical techniques. So this is the some material and method example source of the data information about the statistical analysis. So first of all, before I go to this slide, I wanted to add on this thing that how much information we can add on that our manuscript. It will be helpful for us and it will be helpful for the reviewer as well as the editor. So we don't need to try to hide anything. So this is the article published in Q1 journal that modeling earth system and environment. So if you see this 6 or 7 line, so I have prepared mentioned that which data I have collected, SARC and China, from where I have that collected the data, what is the data source I have mentioned, how much data I have used for the training and model validation I have mentioned, and which statistical package I have used. So within 6 or 7 line, without going to any material and method, anyone can understand that what is the data source, what is data training, testing. So this way we can conclude our information. This is like the second example. This is published in the current research in food science. So here also we have mentioned that we have taken the total pulses production for the major 5 states and India. This is the time and this is my data we have used for the training and testing, and the software we have used the RSpec software. Sometime because most of the time the existing material and method is there. So we can show our material and method like in that way also. So after that, if we are putting this kind of flow chart or schema, we need to give little bit write up about our material and method. So we can mention that what is our database, how much data we have used for training and testing, which model I have used for the forecasting, what is the model selection criteria, what is the validation selection criteria. So if anyone know about this field, so only by seeing this picture he can understand the whole work. So we have to focus on that also. This is another work which is published in the MDPI viruses, where we have taken the January 2010 to December 2010 data, and we have taken the data of the food and mouth disease of the Thailand. Okay, and this model we have selected, and this model we have used, and on the basis of this we have selected the our best model. Now I mentioned that use of good statistical figure instead of table in manuscript. So if you see this table is, I have to read so many things, but if I see this table, so I can understand that all the treatments are statistically different, and clearly we can see the difference also. So sometimes good figures can give more clear picture about the information about the data. So we can use this also. We can use different type of visualization, bar chart, box plot. Now, because the if we are doing the lab study or whatever, so sometimes this is the one example of the any field or lab technique. So how we can compare that which of the treatment is best. So in internationally, the Turkey HSD test is more reliable for the decision making. So instead of using other tests, we have to go for the Turkey HSD test. So like this way, in your field, you can choose that which method is statistically more validated by the international reviewer or journal. So you can follow that also. This is the article we had published in Plant Biology, and why I kept this, because this article when we publish, we have not kept any table, only by the figures we had published our article in Q1 journal. So Q1 journals is very helpful also for okay for our career in growth. So it is not important that every time we are giving the tables, we can put some good figures and give results also. Okay, so we can use that when we are going for the research ethics and statement. So nowadays it is very important that whatever research approval and consent is important. Conflict of interest is a statement we have to give. If you are any editor of the journal, so kindly mention that you are the editor of that journal. So no conflict of interest come later on. Author contribution is very much important. That what is the, because we are in very tough era of research, so we have to give the more information how much we can give. Any funding if you have, you can give. Institutional review board statement, if you are doing any animal study, so you have to do. Data ability also, that how anyone can access your research data, so you have to give that information as well. This is something I can say not new, but nowadays we are, when we are seeing the journal, so this word we are getting too much. Reason for the paper retraction. So after, don't think that you, once you have published the paper, so your duty is over. Nowadays you have to careful before and after also, because sometimes if anyone using the fraud data, fraud, plagiarism, or anyone name is an added without their consent, so they may be come as a unethical behavior, so article may be retract. So you have to careful, that's why I am mentioning whatever information you are using, kindly recheck, take the consent of the all the authors, then only go. Sometimes self retraction is there. If any authors think that my article is any having result mistake, so they can honestly say this is mistake, and paper can be retract due to this misleading result. Okay, so this is also happening sometime, so careful. What editor looks for in manuscript? Quality, definitely. Novelty is there, good science, good presentation, because editor is first stage of your manuscript. So before he send to any reviewer, first he will check, and only directly he sent for the review. So once he will roughly check that, and what is the language, what is the how they have prepared the article, they have followed the journal's instruction or not, that significant and originality, consistency, well written story, and author energy, because it is also important that we have written scientifically, and it is showing the our author energy also during the whole manuscript. In strong introduction, because when we are writing the introduction, so we have to careful, we have to give clear background of study, what is the history, and why we are choosing this study. It should be come properly, explain this, why this work is the study, what is the objective. Clearly logically organized complete method we have to give in material and method. Results should be very clear and properly, tables are explained, each and every figures started, or relevant discussion, it should be meet to our result, and whatever the objective we have added in introduction, it should be properly achieved. So this is the article publishing process. If it is submission and it does reject, then if it is crossed to editor, then go for the referee, referee can reject, then after referee it can go for the revision, then reject, acceptance, more revision. So this is the process. And if sometime it is going for the desk reject so many times, so you have to be careful that what is the reason that article is not going to further review, so that we need to check. And before we submit the article, it is good if we are 4 5 authors, or if you have our mentors or teachers, so we can check internal review. So if we go from proper internal review, then it is very less chances that the article is coming for more correction. So internal review is important. Then write a cover letter and explain the your what is the importance of your study, and take the all consent of the all the authors to submit in one journal, because we have to submit our article one time in one journal. If they are taking too much time and you are don't have time, so just write a mail also, that what is the reason for delay, because sometimes editor is not trying to delay, but if they are not good getting good reviewer, so they may be taking some time. Okay, so that also one reason that our article is quite going to delay. This is the some indexing citation in SCI index, Web of Science. So, if you have any question, you may ask regarding this, because I have to show so many things, but let due to time consent, I was and I just finished as much soon I can do. Thank you so much.
Thank you so much, sir. Most of our participants, they are connected on YouTube channel also. So I
Mm-hmm.
think the question session is not possible here. We will answer
Okay.
their queries in chat session. Thank you
Okay.
so much, sir, for your great session.
Yeah. Thank you, ma'am. Thank you.
Sincerely thank you, Dr. Pradeep Mishra, sir, College of Agriculture, Riva, for his insightful and informative expert talk. His valuable guidance on research publication process and identifying reputed journals will definitely benefit our researchers and young scholars. We are truly grateful for the time and knowledge and the inspiration given by Dr. Pradeep Mishra, sir. And thank you, sir, for your precious time given.
Yeah. Thank
Now,
you.
now, in the next session, I warmly welcome our another invited speaker, Dr. Mahi Chore, Department of Biotechnology, AKS University, whose work in biotechnology contributes significantly to advancing scientific knowledge and innovation. Her research domain focuses on microbiology, particularly plant growth promoting rhizobacteria and metagenomics, which play an important role in sustainable agriculture and soil health management. So now I welcome Mahi ma'am to please proceed with the session. Yes, ma'am, you can proceed with. Thank you.
Thank you so much, ma'am. Good afternoon, everyone. First of all, I want to extend my thanks to organizers for giving me the opportunity for the presentation. And now I'm going to start my presentation. I'm sharing my screen. Is my screen visible?
Yes, ma'am, the screen is visible.
Okay. So you can see the title I have mentioned, Metagenomic Exploration of the Indigenous PGPR for Organic Agriculture. So firstly, let me tell you a little information about my area of the, actually, I have performed my research work on plant growth promoting rhizobacteria and their microbial assessment for organic and inorganic agriculture practices. So I'm showing my first slide. For better connectivity, I'm going to off my video.
Ma'am, at the bottom.
Yes.
Yes.
My screen is visible.
Yes, ma'am, you can continue. Slideshow mode, yes, definitely, perfect.
Okay. So I have shown my first slide. In my first slide, you can see some introduction I have given about the chickpea. That is my crop. I have worked on it. So firstly, I want to tell you why this crop only. Actually, India is largest producer, consumer and exporter as well of the chickpea crop because of its nutritional value. And it has two varieties, Desi and Kabuli. And India, Desi is more common than Kabuli. Desi type has small angular seeds with thick seed coats and color is quite brown or dark. And Kabuli type has light color with thin seed coat, that is why it is more digestible. Here in the slide, you can see some infections of the chickpea are given by fungi, bacteria, and virus as well. Here is name of diseases are given, like Physidium wilt, in which wilting occurs in the leaf of the plant. Ascochyta blight, in which dark color develops in the seed coat, and botrytis gray mold, in which grayish color develops in the pods. Similarly, more diseases are given here. Biocontrol PGPR agents are also given. Here, some PGPR activities are given, which are actually performed by some bacteria like Azospirillum, Bacillus, Pseudomonas fluorescens, Bacillus subtilis, and more. Some techniques to study plant growth promoting rhizobacteria associated with chickpea plant are given here, like 16S-RNA gene sequencing, metagenomics, and DGGE, TRFLP, and so on. Okay. So I am diving in my topic in depth a little bit. So you can see after metagenomic assessment, proteobacteria, actinobacteria, firmicutes were found predominant. And because of, in the I'm going to show the benefit of metagenomic assessment actually, because by culture dependent technique, we need to collect the sample, then microbial isolation, followed by screening of microorganism. Then we need to perform some biochemical characterization test for the identification of microorganism. Then we can do pod trial or field trial. After that, we are able to make biofertilizer. And later on, it is not necessary we'll get good result. But in case of metagenomic assessment, this is culture independent technique, which is time saving, and we can get directly detailed information of the microbial diversity related with the chickpea plant. Then we'll be able to make biofertilizer in short time period, and there is more chances of the effect of biofertilizer, which is prepared by using this method. So coming back, okay. Here you can see some metagenomic some points are given about the metagenomic techniques. Then we'll discuss in detail later. Besides those microbial diversity, proteobacteria, actinobacteria, firmicutes, bacteroids were also very dominant in rhizopheric area of the chickpea plant, which are actually involved in playing major role in the growth and development of plant. Here, metagenomics overcomes culture-dependent limitations that just I have explained, limitations to reveal full diversity of rhizospheric and endophytic microorganisms, and it identifies unculturable taxa crucial to plant health and productivity, as well as it highlights high microbial richness in chickpea rhizosphere, and more properties it is not possible to explain in detail because of shortage of time period. So I'm directly coming to the point here. Firstly, for quality control, by using program trimeomatic, low quality reads are trimmed. Later on, by using databases like Silva, Green Genes, and RDP, bacterial taxa are known. Later, after that, followed by a functional annotation of genomic assessment of the microbial diversity. So some more microorganisms are given that were assessed by metagenomic assessment, like Burkholderia, Pseudomonas, Rhizobium, Mesorhizobium, Bacillus, Clostridium, Actinobacteria, Ascomycota. For example, Rhizobium is quite fast grower, Mesorhizobium is quite slow grower. So by culture dependent methods, it is not necessary we'll get Mesorhizobium growth also. So metagenomic assessment provides details of slow grower microorganisms as well as which are culture independent too. And these organisms are involved in plant growth promoting activities like nutrient uptake, stress tolerance, nodule formation, biocontrol of pathogens, antibiotic production, mycoparasitism, biocontrol activity. So my objectives were firstly collection of the soil sample from two regions of Madhya Pradesh, Satna and Chhindwara. I had collected organic and inorganic soil. Then after that, my soil samples were introduced to the metagenomic assessment, and after that, relationship between those microorganisms which were indigenous to the chickpea plant were found. These are the name of samples, and total 16 samples were collected from two regions that just I had told, Satna and Chhindwara region, inorganic and organic soil, as well as diseased and healthy plants were also collected. So in this stack bar plot, you can see 30 taxa at the genera level. These 30 taxa were very dominant at the genera level. Later on, these genera were studied in detail at species level too. So in species level, you can see Pseudomonas species like Pseudomonas putida, Pseudomonas fluorescens, and Pseudomonas other species. These were very dominant. And Pseudomonas is basically involved in biocontrol activity of the plants, leguminous plants or other plants also. And they were very dominant in organic soil in comparison to inorganic soil. Same data were analyzed by heat map also. So you can see the dominance of Pseudomonas in all samples by the dark color in this heat map, followed by Pentova, Acinetobacter, Enterobacter. These are very dominant. Then same dominance was studied in this were studied and showed in this heat map at species level of the top 50 taxa. Okay, in this detail points, you are able to see that this microbial divergence is because of the location. Because Chhindwara and Satna, there is a distance, there is a huge distance between these two regions. So because of the different locations, diversity, richness, and diversity evenness were also found different. So you can see. Okay, in this third and fourth point, I'm going to explain you about the core bacterial phyla and functional relevance. Proteobacteria dominate all samples, forming the core microbiome across both the locations. And proteobacteria like actinobacteria and other firmicutes, bacteroids are comparatively enriched in organic system of the Satna soil, basically. Because inorganic farming was more dominant in Chhindwara region in comparison to the Satna. Here, this is showing this figure is showing about the presence of microbial diversity, and these were indigenous in the plant root area and in the rhizophoric area as well, and they were involved in actively plant growth promoting activities. And by showing these activities, these microorganisms were enhancing the growth of chickpea plant, and these are Cosaconia, Streptomyces, Enterobacter, Anthomonas, Bacillus, Bradyrhizobium, Pentoa, Streptomyces. So overall, the findings clearly demonstrate that organic farming practices support diverse, stable, and functionally active bacterial community location-wise. So PGPR-based biofertilizers and biocontrol agents are more dominant. In this alpha rarefaction curve, you are able to see at age, tau, and observe level the increase or raiseness, the increase and raiseness in the all microbial diversity, and at Shannon level, there was more evenness in the microbial diversity. So increase and evenness both were found in organic soil. Now this stack bar plot is showing the relative abundance of top 30 taxa at the genera level, and it includes Fusarium, Coletotrichum, Aspergillus, Cercospora, Arvenia. These were very dominant in organic soil samples, and these are not very pathogenic, but among these few are very pathogenic for the chickpea plant. For example, Fusarium, Coletotrichum, they are more dominant in developing infection in the plant. In this bar plot, top 30 taxa at species level are given, like Physarum. Just I have said that Physarum is very dominant in development of infection. In previous slides, I had shown you the infections developed by Physarum and other fungus too. So they were very dominant in samples which were collected from the inorganic soil and disease soil also. Same presence of pathogenic fungus is shown in this heat map. You can see dark colors dark colors showing the presence of Fusarium, Pyrrhocalaria, which are very dominant fungus. Similarly, this heat map is showing the presence of species of infectious pathogenic fungi, which were very dominant in the inorganic soil samples, and they were involved in development of various diseases in the plant. So in contrast, inorganic and diseased soil showed increased abundance of pathogenic fungi that I had shown about the Fusarium and Colletotrichum and other fungus too. The dominance of these taxa corresponds with reduced fungal evenness and indicates microbial imbalance, which is commonly associated with higher disease incidence. So, and in contrast, organic agriculture practices foster beneficial fungal consortia that naturally suppress soil-borne pathogen, as well as enhancing soil health and crop resilience. Besides these work, functional annotation of plant growth promoting genes under organic and inorganic conditions and diseases soil were also performed, and you can see KO and protein family analysis of plant growth promotion and disease suppression in plant. Organic and normal rhizosphere or soil samples showed highest enrichment of plant growth promoting gene and biocontrol related KOs and PFAM analysis. And in contrast, inorganic and diseased soil sample shows lowest enrichment of plant growth promoting genes and their activation. Same data I have shown here with the help of heat map, so it will be easy to understand. This is a heat map for the protein family analysis expressed by genes. So overall inference include organic rhizospheric soil harbor the most functionally competent microbiomes for plant growth promotion and disease suppression, followed by organic normal plant samples. Whereas inorganic and diseased samples show functionally depleted microbial communities, resulting in low quality or diseased plant as well as their pods also. This heat map same is showing all PGPR genes. Organic samples show the highest abundance of PGPR genes, and while inorganic samples show reduced presence of PGPR gene abundance in crop. And this is heat map which is showing all biocontrol genes present in the microorganism which were present in organic soil and healthy plant samples. So in this you can see high expression of chitinolytic genes, cytophore biosynthetic genes, antimicrobial genes were highest in abundance in organic soil samples, and comparatively inorganic and disease samples exhibit down-regulated expression of these biocontrol genes. So coming to meta-analysis based case study, this is showing, actually I am showing this data which I have collected from after studying various research papers. So in those research papers from where I have collected data showing the microbial evenness and microbial divergence in organic soil samples, comparatively in inorganic soil samples, there is clumping in the microbial diversity, and which were clumped together and not actively involved in plant growth promotion of any crop. Same details I have shown here with the help of heat map. You will be able to understand heat map. This is this dark this dark color is showing the presence of plant growth promoting activity in organic soil samples, whereas in inorganic soil samples, there is less expression or low plant growth promoting activities were found. In this, you can see this green color line. You are able to see this is for the organic soil samples and plant growth promoting rhizobacteria, which is highest in abundance, whereas in inorganic soil samples, this is lowest.